technology now. So, let's see if this works. You know how many times I have trouble with this, so God only knows. Called Gene Transfer. It's in the Unseen Life on Earth series, so you can't see it online if you wish.
Called gene transfer.
Yeah.
Because I chapter 7.
I want to throw this thing out the window. Anybody have a window? Okay, I am prepared for this. I have 4 copies. This machine loves giving hell.
I'm not fun.
Now this one needed 8 last year, so at the beginning there's a little This goes in your lab record book, front to back, under Unseen Life on Earth, and then transfer, and nothing. Would you hit the VCR button over there again? Nothing, right? There it is. Now it's going fast.
What's the name of this video?
Yeah.
Unseen Life on Earth.
Nope, that's the series.
Gene transfer.
I wrote it on the board, I said it 5 times. Think about it.
Do you just pull it up on your site on your
Yeah, but it's really small and not so good. But I can get this one. I have one very good and very bad trait. One is I never give up. Ever.
We're watching this, and then we're also watching the
Yeah.
mouse that landed on.
But not at one right after the other. We can go later. Now it starts. When I threaten to throw it on the floor. Okay.
Dr. Hicks, is there, never mind.
Down deeper, I want to do.
Is there what?
Never mind.
And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
It's no secret that genetic information is passed on from generation to generation. I mean, take a look around. The passing on of DNA in plants and animals is achieved by a mixing of genetic information, which occurs as a result of reproduction. And it's easy to see the genetic traits that organisms share. Microbes pass on genetic information too, While their sex lives are quite different from ours, their DNA is nonetheless successfully regenerated. Beings, along with other complex life forms, transmit their genes only to subsequent generations. So-called vertical gene transmission means that genetic information is passed on only through the reproductive process. Microbes do this too, but in addition, they have another extraordinary ability that is a far more powerful transmitter of genetic traits. I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore the three main mechanisms for microbial genetic recombination. That's transformation, transduction, and conjugation. And we'll also learn how humans have harnessed these mechanisms and applied them for the benefit of all of us. Unlike you and I, or any other creature you can see here at the fair, microbes can pass their genes not only to their offspring, but also laterally to other microbes of the same generation. This is known as horizontal gene transmission, and it's a trait that really sets the microbes apart from multi-celled life.
More than one pilot.
To learn more about horizontal gene transmission, I'm here at Oregon Health Sciences University to meet with Dr. Maggie So, chair of the university's Molecular Microbiology and Immunology Department.
I'm interested in how these microbes can acquire virulence traits. And many of these characteristics can be acquired, can be rapidly spread among a population of bacteria through these mechanisms. The microbes in your body might be totally harmless until they acquire these traits through horizontal transmission. And by acquiring these traits, they can suddenly become more virulent. They can acquire drug resistance, for instance. So if they cause disease later, they would be harder to treat.
So is there a disease that's a good example of this horizontal transfer process?
Well, a good example of this is diphtheria, which is a disease we don't see anymore because of childhood immunization programs. The disease is caused by a bacterium called Corynebacterium. And for the bacterium to cause disease, it has to produce a toxin, which kills the cells in your body. And the gene for the toxin is transmitted to the bacterium via a bacteriophage. And a lot of toxin genes are actually transferred to bacteria through the process of phage conversion.
Phage conversion can result from transduction. Genetic material passes from one bacterium to another via a bacterial virus called a bacteriophage. The virus injects its DNA into the bacterium. The bacterium then becomes a virus factory, replicating the viral genome, making viral coat proteins, and eventually releasing hundreds of new bacteriophage.
Every once in a while, during replication of the viral genome, a small piece of bacterial DNA gets inserted into the coat protein as well. And the live virus now transfers this genetic information from the recipient bacteria to a new bacterium.
In conjugation, another form of genetic transfer, a donor cell comes in contact with a recipient cell by way of a conjugation tube known as the sex pilus. When contact is made, a single strand of DNA containing the genetic information goes through the pilus from the donor cell to the recipient cell. This single strand of DNA is usually in the form of a plasmid, which is an extra chromosomal, independently replicating piece of DNA. A second strand of the genetic information quickly gets synthesized in both the donor and the recipient cell.
So ultimately, both cells now have complete copies of the genetic information that was transferred.
So what's the significance of that from the perspective of someone studying disease, such as yourself?
Well, we now know that a lot of genes coding for drug resistance, antibiotic resistance, reside on plasmids. So a single bacterium in a population harboring a conjugated plasmid containing an antibiotic resistance gene can ultimately spread this genetic trait through the entire population of bacteria, and this could happen quite quickly in the lab.
In transformation, a wide variety of bacteria have the ability to bind free-floating DNA from their immediate surroundings. This DNA may then be inserted into the chromosome and become a piece of permanent genetic information. Bacteria that acquire DNA in this manner inherit the genetic traits that are encoded in the incoming DNA.
How is it that there's free-floating DNA in their environment?
Well, many bacteria have, in addition to the growth cycle, a lytic cycle. That is, they die, their cells burst, and their DNA is released into the surroundings. And living bacteria that are in the neighborhood, if they're competent for DNA transformation, will then take up this DNA. Now, there are bacteria that are very indiscriminate in the kind of DNA that they take up, such as streptococci. And then there are other bacteria that are more particular, such as Neisseria and Haemophilus, and they'll only take up DNA from their own species.
There is another highly specialized form of DNA transfer known as transposition. Pieces of DNA called transposons hop from one genome to another, and sometimes from one region of a genome to another region of the same genome. These self-sufficient transposable elements may code for antibiotic resistance.
So let's say we have a transposable element that codes for a drug resistance gene. This transposable element hops, say, from the chromosome to a conjugated plasmid, all within the same bacterium. This bacterium now makes contact with a female cell, and the conjugated plasmid gets passed to the female cell. Well, the transposable element now ultimately gets passed to a second bacterium via conjugation.
Okay, so it's sort of a two-step process.
That's right. A transposable element can't transfer itself from bacterium to bacterium. It always needs an intermediary.
Knowledge of how bacteria transfer their genes can be used for our own benefit. For instance, not only can we transfer genes between bacteria, but similar processes are used to transfer genes into eukaryotic cells, like plants. And it turns out that some of this high-tech genetic manipulation is happening in some very unlikely places. This is the village of Chunamina in Zimbabwe, southeastern Africa. There has been no rain for many weeks. The village's crops of corn and millet have failed. The only staple left is cassava, its starchy, edible tubers safe underground, protected from the scorching sun.
In Africa and South America, that's what they replace potatoes.
Victor Masona is a Zimbabwean plant biologist.
Cassava is the only standing crop. When we dug up a few tubers, I mean, it was like everybody was going for them. even before they were cooked, which means there is evidence of hunger. Everyone is hungry.
Cassava has brought Masona to this village. The plant is eaten every day by 500 million people in Africa and around the world. Westerners seldom encounter it, except in tapioca pudding. But here, it is the difference between survival and starvation. When something goes wrong with this crop, there's nothing left to fall back on. And something is wrong.
So this is the African cassava mosaic virus. You know, the disease that affects cassava, and you can see it by the crinkling of the leaves. You know, there's a mosaic on the leaves. All the shoots that come out have got the same disease. So you can actually spread the disease by propagating cuttings from this plant. So it can also be spread by whiteflies, sucking on this plant, and then they go and suck on to the next plant, and that way they transmit the virus from one plant to another.
Masona's collaborator is plant biologist Ian Robertson of the University of Zimbabwe.
Throughout Africa, this material is dug out of the ground. They see these symptoms, but they don't know it's the virus. So they'll break off pieces
Very easy to plant. Stick it
and they'll pass them to their relatives. And they'll plant them, but they'll plant them with the virus in the stems. So when the thing grows, it'll be full of virus, even fuller than this. And the difference between this plant and this plant is this one will give you at least 30 tons per hectare, and this one will only give you 6 or 5 or 4 or 2.
The villain is the tiniest of all microbes, a virus. Viruses are parasites. They enter animal and plant cells and use their victims' biological machinery to reproduce.
That's actually HIV right there.
Viruses are difficult to fight. Pesticides could help defeat this virus by eradicating its insect carrier, the white fly. But chemical treatments are too expensive and also might contaminate the water supply. Victor Masona uses another line of attack, one that will genetically alter the cassava plants.
Our work involves the genetic transformation of cassava for virus resistance. What this means is that we are trying to introduce genes into cassava. We hope that at the end of the day we will have genetically transformed cassava which is resistant to African cassava mosaic virus.
If you get the gene in, then we can produce for them Victor's kind of stuff, which is going to be virus resistant. Then we begin to solve the problem.
Masona obtained a grant to pursue his experiments at the International Laboratory for Tropical Agriculture Biotechnology in La Jolla, California. He fights one microbe, the cassava mosaic virus, with another microbe called an agrobacterium. Agrobacteria routinely pass their genes into plant cells. In a test tube, Masona mixes a solution of agrobacteria and genes from the virus. An electric current punches holes in the agrobacteria. The mosaic virus genes enter. The agrobacteria then transfer the viral genes to cassava plant cells.
You're trying to vaccinate the plant. Right? So we're putting a gene from the virus into the plant, so it becomes part of the plant.
Masona gives the plant just enough of the virus so it can defend itself when the real virus attacks.
And the agrobacterium transfers the gene to the cassava cells, and then we take the cells, culture them, and grow them into plants.
After 5 months of careful nurturing, Victor has a finished product. A generation of genetically altered, mosaic virus-resistant cassava plants. The next step is to field test the transgenic plants in Zimbabwe's climate, with Zimbabwe's farmers, among Zimbabwe's whitefly population. Ian Robertson dreams of the day when Masona's plants will thrive throughout Africa.
I'll go through all this procedure of multiplying in the field, and in 3 years' time we'll have 200,000 for the next batch of people who need it. Then they'll get 30 tons per hectare every year, and their cousins will get it, and their friends will get it, so suddenly we'll have 30 tons a year, more or less permanently.
But it's not quite that simple. Genetic engineering is a powerful new technology. Its long-term effects are hard to predict. Zimbabwe's leaders are cautious.
Still has not been approved.
They're drafting regulations for the testing of transgenic plants. Masona cannot bring his transformed cassava into the country until the regulations are in place.
It can be very frustrating that the plants are sitting in my lab in California. I cannot proceed with the testing and the introduction of genetically engineered cassava if the regulations that guide these sort of introductions are not in place. So the people are hungry, but there's something that could help the situation, and yet I'm not able to bring it here because I'm not allowed to do so.
Biotech holds out the promise of enormous power. We can manipulate crops, animals, and even people. That's a dangerous power. So we need decent people to make the choices.
Scientists like Masona are sometimes accused of toying dangerously with Mother Nature, of trying to play God.
I don't think I'm playing God. I think if God would even be happy about that situation, because our aims are to help alleviate the problem of disease in cassava in this case, by obtaining crops that can be used by very desperate people. And I don't think that is bad.
Masona hopes he will soon be permitted to test his cassava plants in Zimbabwe. If they prove resistant to the cassava mosaic virus, a young biologist's manipulation of the power of microbes could nourish hundreds of millions of people.
To you is that plants or crops can also suffer from diseases. My research involves trying to put things into the crop so that, or into the plant, so that it doesn't suffer from disease.
Perhaps Masona's attempts can only be successful if 21st century science is brought to everyone through education. When genetic transfer processes like transformation, the one he uses in his work, are better understood by all of us. Experiments with DNA transformation are also conducted regularly by Dr. So and her staff at Oregon Health Sciences University.
We do DNA transformation quite a lot in the lab, and we use the technique to genetically manipulate our bacteria, create mutations that we can follow for our studies, put in new genes that we can test for their virulence characteristics. So I can give you a demonstration.
Perfect.
I've streaked out a strain of bacteria onto a plate, and these have been grown overnight. I'm going to swab these cells into these tubes and add DNA to each of these tubes and incubate bacteria with DNA and let the bacteria bind and take up the DNA. Now, the DNA contains a drug resistance gene, which has been inserted into a gene that we think might make our bacteria virulent. So we're actually mutating the virulence gene by inserting the antibiotic resistance gene right in the middle of it. The bacteria take up this DNA and insert it into their genome. They're going to be resistant to this antibiotic. These plates contain antibiotics, so bacteria that have taken up DNA will be able to grow on these drug
Bacteria
plates.
shouldn't grow unless it picked up the resistance.
So after an hour of incubation, I've taken the bacteria and I've spread them on antibiotic medium, and then I've incubated the plates, and that's an overnight incubation. And here are some plates from yesterday's transformation experiment.
Okay.
First of all, we have the no DNA control. The bacteria were not incubated with any DNA, and so you see there's no transformation. They can't grow on drug plates. Here we have bacteria incubated with a large amount of DNA, and you can see that the plate is covered with bacterial colonies.
So they inherited a great deal of antibiotic resistance.
Well, that's right. The higher the amount of DNA, the more transformation events that take place. So a larger population of bacteria have now acquired the genetic trait. Here's a plate containing the smallest amount of DNA, and you can see that there are bacterial colonies on the plate, but there are fewer of them. We are using genetic engineering to try to understand how bacteria cause disease. In addition to that, scientists are now perfecting processes of DNA transfer to higher organisms based on these age-old bacterial genetics techniques. So in the case of farm animals, it is hoped that gene therapy can improve animal husbandry. And in humans, it is hoped that gene therapy can be the answer to a wide variety of medical problems.
For instance, we used to get the insulin used by diabetics from pigs like old Prince here. Now, pig insulin is close to human insulin, but not exact. Now, we can make human insulin by using microbes. By understanding the mechanics of microbial genetics, we can manipulate those mechanics for our own benefit, from improving health to turning the tide on world hunger. Over the 3.8 billion years they've been evolving and coping with changing environments, microbes have developed a variety of mechanisms for passing on genetic information and for survival. In the next episode, we'll see that it's a skill microbes have been honing since the dawn of life.
I know, they are. Could you hit, I think it's laptop button. Okay, so one of the things that you saw as a problem on this was the different points of view about manipulating genes. And there is the most bizarre dichotomy here. In Europe, you can do research on anything in genetic engineering. There are no limits, stem cell limits or anything like we have here. But no matter what you discover, it can't be sold or planted. Africa has adopted mostly the European attitude. Now in the United States, we have the other radical point of view, and that is anything they limit research. You can't do it on stem cells, you can't do it on this cell line, you can't do it on that cell line, but there is no limit on what you can sell or stick in the ground, and here's the biggie, it doesn't have to be labeled. And so this is upsetting to me and a lot of people because I'm not afraid of genetically engineered tomatoes that won't rot as quickly as the tomatoes now. They put a fish gene in the tomato. And the reason is, what's the job of a tomato? It's a fruit, and what do fruit do? They have seeds. They rot, they drop the seeds, the plant grows. It's reproduction. So the moment the tomato is ripe, rotting gene is triggered to release the seeds. Well, we like to eat the ripe tomato. We don't want it to rot. So the problem is you can't pick it when it's ripe because it starts rotting. So what we do is we pick it when it's green and hard. Then we put it in pasteboard so it won't get bruised, and we ship it. But it has the taste of what it ripens in, so it tastes like pasteboard. I saw my mom on Skype not too long ago, and she was eating this huge beefsteak tomato sandwich. She was eating a BLT, and I said, "Man, that looks good." And she says, "Well, that's not that great." And I said, "Why?" And I said, "That tomato looks wonderful." She said, "It's wet and it's red, but it has no taste whatsoever." Because they pick the varieties to plant that ship well. They want to look good and they want to ship well. They don't care what they taste like. And they pick them green and ship them. Now, so what Monsanto did was they knocked out the rotting gene, and they could have put any gene in there. They put a fish gene in. And now, when it gets ripe, it triggers the rotting gene, but there is no rotting gene. So they can pick it ripe, and so long as they don't bruise it, it doesn't begin rotting. And so it's sort of like the sterilized milk that you get. You buy organic UHT milk, ultra high temperature. That milk is sterile. The milk you buy in the gallon has about 1 million E. coli in it per mil. Why? We make 1 million pounds too much milk every day. And the dairy farmers, you know how we say we believe in free trade? We really don't. We say, listen to the dairy farmers. We know that if you really had to compete, milk would be 50 cents a gallon. You wouldn't make a profit. You'd go out of business. So we're going to guarantee you $3 a gallon. So if you're the government and you're guaranteeing the dairy farmers $3 a gallon, do you want milk to spoil every 10 to 14 days, or do you want it to be sterile and last 5 years? You want it to spoil. That's why we use low temperature pasteurization in this country. If you go to Brazil or any other country, milk is not refrigerated. It's in a paper container, it's setting on thing, and it has a 5-year expiration date. Why? It's sterile. Things don't rot unless they have bacteria in them. And if you put sterile milk in a sterile container, it will last 5 years. And by the way, it tastes a lot better. And I don't like drinking millions of bacteria. So I never buy regular milk here. I always buy the UHT, and you can always tell because it costs a little bit more, and they say organic on it, and then it'll say UHT, and then you'll look at the expiration date, and it's 3 months once you open it. It's just, and people go, ew, think about it. What's evaporated milk? The same thing. Sterile milk in a can. Until you open it, it's sterile. So anyway, in the United States, the Bush administration said that farmers do not have to label genetically engineered food. Now, in America, I thought we were supposed to have a choice. And I don't mind genetically engineered food. I don't think it's any different than anything else. And I don't think it's harmful. But I want people that don't want it to be able to make a choice. And the fact that they don't label it annoys me to no end. But I still buy those genetically engineered tomatoes because they actually taste so much better. But they also cost $2 a piece. So anyway, we're going to be talking about moving DNA. Make sure you know the attitudes toward recombinant transgenic organisms. They can do any research they want in Europe, but they can't plant it or sell it. Here, we limit the research, because of all the moral people, and we don't label it. So anything can be sold, and they don't have to label it. All right, so we're talking about DNA, and we're talking about moving DNA. So the questions I'm going to ask you, first of all, I want to let you know that Ventnor, anybody know Ventnor? He's the man that sequenced the human genome, and he did it in 3 years. There was a big competition to see who would first sequence every gene in the human genome. And he developed a whole new technique called shotgun sequencing, where he labeled each base with a different color and was able to sequence the human genome 2 years before his competition. And so now he's the first person who ever developed a synthetic life form. He took a mycobacterium, knocked out the inside, and added genes from what he wanted to. And he made a microbe that he named Mycobacterium laboratorium. And its job is to make ethanol biofuels from garbage. So it's the very first synthetic life form, and it's called, when you do this, he's not creating life, remember, he's using pieces from different organisms and putting them in a mycobacterium after knocking the inside out of it. But it's called synthetic genomics. So that's a vocabulary question. What's synthetic genomics? Taking pieces from different microbes and organisms and making a totally synthetic, reproducible bacteria. All right, so we're going to be talking about natural gene transfer and unnatural gene transfer. Natural means it happens in nature. We've already talked about them, so it's just going to be a review. It's transformation, transduction, conjugation, and transposition. And then there are the other two that occur rarely. And remember endosymbiosis, where the mitochondria and the chloroplast became a part of the eukaryotic cell. And rarely in acidic conditions, two bacteria can be forced to join each other when their cell wall is digested away, and that's called gene fusion. And it does happen in nature. So all of these happen in nature. Now, by being pervs, standing behind the door and watching them do it, we figured out how to do it ourselves. And this is unnatural or genetic technology. And so this electroporation, what he used, knocking holes in bacteria so it would absorb genes, that's artificial transformation. By watching this, they learned to do that. And almost all of the hybrid plants that are disease resistant are the result of electroporation techniques, where you're basically vaccinating the plant. All right, so remember the two differences that we talked about. What's the difference between genetic recombinant DNA technology and transgenic? If you're using a eukaryotic gene into a prokaryote, it's recombinant. And it doesn't work well because they don't do post-translational modification. What do they do now? They put a eukaryotic gene in a lower animal. They take a human gene and put it in a sheep or a goat or something like that. It makes factor 8, factor 9, anti-clotting, all of that, and that's called transgenic DNA technology. And we don't use the word DNA engineering anymore. Anybody know why we don't use engineering anymore? Engineers work with machines. You want to treat living things like machines? I mean, when my microwave breaks, do I take it to the microwave repair shop? No, I throw it in the garbage and get another. That's the way Americans do it. That's the way they treat their machines. Do we want to treat living things that way? No. So there has to be a moral component, so they've gotten rid of the word genetic engineering, and it's called genetic technology. All right, so I'm going to go over the natural ways of moving DNA briefly, and these are the questions you must answer from this chapter. Yes?
Could you go over recombinant and then transgenic? I know that you said recombinant, they don't do post-transitional remodification.
Yeah, that's where you
And
take a gene from a human and you put it in E. coli.
Okay, gene from human into E. coli. And then what did you say the other one was?
You take a gene from a human and put it in another eukaryotic animal, like a goat or a sheep or a cow.
What's that one?
Transgenic DNA technology. You're transferring a eukaryotic to a eukaryote. Okay, so here's what the questions are going to be in this chapter about is it natural or unnatural? If I give you a mechanism, you have to tell me if it's natural or unnatural. So if I say electroporation, what is it? Unnatural. Okay, what if I say transformation? Natural. So you got that one. How much DNA is moved? It's very simple. 1 or 2 for transformation, transduction, and everything, but conjugation, 51. Now, Artificial transformation can move 100, but that's not natural. So, remember all the others, transduction, transformation is only moving 1 or 2 genes. But conjugation, when you're transferring that whole plasmid plus 1, can be 51 genes. Can be 51. All right, so first question is, what or how moves the DNA? And of course, in transduction, it's a virus moving. Yeah,
That, what is it called? That's when you put the hole in it?
that's electroporation.
Electroporation, that's only 1, 2?
That is not natural. It's unnatural. It's artificial.
But how many? Because that's
Oh,
the
it could
question.
be 100.
It could be 100.
Yeah. But I'm not going to ask you that because it's artificial and it's, you know. So each one of these things that we studied in nature led to a revolution in science. So I want you to know which experiment led to what revolution. We're going to go over it. And then finally, we're talking about bacteria. Which of these, if any, had an effect on evolution? If you're moving 1 or 2 genes, you're not affecting evolution. But if you're moving 51, you are. So the gram negatives, remember, only gram negatives do conjugation. So conjugation has had a major effect on the evolution of the gram negatives. All right, so we're going to go answer these questions about each one of the 3 times. And the first one is transformation. Remember, this is Griffith. He said, I want to make a vaccine for pneumococcal pneumonia. And there are 2 subspecies of pneumococci. One has a capsule and one doesn't. Which one will kill you? One with a capsule. So he said, I don't want to make a killed vaccine because killed vaccines wear out after 5 years and you need a booster. I want to make a combination of a killed and a live vaccine because live vaccines are forever. If you take your polio vaccine, you never have to take a booster because polio virus is living in your intestines for the rest of your life. But it can't cause disease, it's an alternate. So he decided he wanted to kill the deadly one and mix it with live harmless one and make a super vaccine. He knows this was dead because he injected it into mice and they didn't get sick. He knows this one was harmless because he injected it in mice and they didn't get sick. He mixed the 2 together and the mouse died. And he was confounded and confused. And he published a failure. This is one of the big points. This experiment was a failure. He didn't make a vaccine. He failed. When he looked at the mouse that was injected with the deadly and the live harmless, he found living deadly. And except for The Walking Dead, dead things don't get up and move. So how did something that was already dead, and he knew it was dead because he tested it, become reanimated? And the answer was, when you die, cells fall apart. DNA is released into the fluid and absorbed by a close relative, and then the gene from that, for the capsule, was transformed or transmitted to the new microbe. So, who explained it? This is very important. Rosalind Franklin. Rosalind Franklin was the woman gypped out of the Nobel Prize along with Hershey. Hershey was the one that x-rayed DNA to get the double alpha helix. Rosalind Franklin was the one that determined from this experiment that DNA was the inherited material, not amino acids. Now, because she communicated by letter with Watson and Crick, and because she died of breast cancer before they made their great discovery, and her lab burned, they would say, well, we really don't remember all of her letters saying that DNA was the genetic material, and we really don't think she should be on the paper, so we're just leaving her off. And Hershey He's the one that x-rayed DNA and gave them the double alpha helix, and they left him off the paper. And for many years, people would go and say, you know, don't you feel screwed? Watson and Crick used your information and got a Nobel Prize, and you didn't even get on the paper. And he said, well, that's all right. I got one for transduction. He got his own Nobel Prize. So they asked him if he was bitter. He said, no, everybody knows they stole it from me. Why should I be bitter? I got my own Nobel Prize. Screw it. So remember, there's always all kinds of this drama going on among scientists. They would kill their mother for a Nobel Prize in a second. Bye, Mom. Yeah. So anyway, why is this experiment one of the 10 greatest in all of history? Because it taught us that DNA can be moved while you're still alive. That you can gain genes while still alive, bacteria. So it led to all of recombinant DNA and molecular genetics. Okay, so it does, and it happens in nature. It happened in his experiment, and it happens in nature. And here's what happened. Remember, he gave the heat kill, and the mouse was fine. And he gave the live one that wasn't heat kill, the mouse died. Took the harmless one and injected the mouse was fine. Mix the two, mouse dies. Take a blood sample, and there you have the capsule, even though you know it was dead. So that was the experiment. And here's what happened. When cells die, they fall apart, and their pieces of DNA float around. And close relatives can absorb. And if they absorb, they will express the DNA. Okay, so what's the importance? The experiment was a failure, but it was one of the greatest experiments of all time because it proved DNA was the inherited material, and it said that DNA could be moved. Before that, we thought DNA was too small and it'll never be moved. We'll just never get that well. Now we know that not only can it be moved, but it is being moved naturally in nature. So if we stand behind the door and peer and lurk and watch it, ooh, dirty, 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 nasty, nasty, we can learn how to do it ourselves. So this experiment led to all recombinant molecular genetics. So two big things. It proved that DNA was the inherited material, and it proved that DNA could be moved within a current lifetime horizontally. Now, how much DNA is moved? Very little. 1 gene. Now, why do I have 20 over here? That's what people that don't come to class, they're going to read this overhead and you're going to say 20. But this is artificial transformation. This is natural. This is just a screw mold because they didn't come to class. All right, so what's required? They have some really weird requirements for transformation. Yeah?
I'm just a little bit confused about if I get the natural one.
Natural is about 1 gene.
How 1
1 gene. And artificial is 20.
So when the gene is artificial?
No, artificial transformation, electroporation.
Oh,
The very first one moved 20 genes.
the one that they electrocuted.
So the
Right.
plasma. So
Not holes in the plasma. Remember Americans, we couldn't get the gene in there. Couldn't get the gene inside the bacteria. So what do Americans do? We shoot a hole in it. We don't do anything. We don't mess around. We don't watch it or anything else. We just blow a hole in it. That's the way we do things. All right, so anyway, look at this. There are stringent requirements for this happening in nature. One's got to die, and one's got to be in a lot of growth. And then one has to produce this weird protein called confidence factor that allows DNA through the plasma membrane. So it doesn't happen a lot in nature, but it does happen in nature. But it has some very strict requirements. They got to be closely related. One of them's got to be dying. One of them's got to be in log growth. And one of them has to make confidence factor. And what does confidence factor do? Allows DNA to pass the plasma membrane. Okay, so artificial transformation is moving genes inside a bacteria from the environment by just blowing holes with electricity. And then you can put, I think there's been over 100 now. The first one they put 20 genes in. And what is artificial transformation called? Electroporation. You're knocking a hole in the plasma membrane using electricity. It was so funny when I was going to school, this was just the cat's meow. The newest thing. And everybody was doing their research on it. Now you can order a $50 kit from a company and do electroporation on the lab in like this one. That's so upsetting. You know, here people are making their entire reputation of learning how tiny bits of electricity, how small it takes to knock a hole in it and not burn it up. And now you have a little lab kit that an 11-year-old can do. Very annoying. Okay, transduction is when a virus moves genes. And it was discovered with a E. coli virus called a bacteriophage. And what bacteriophages do is they attach to a cell and they inject their DNA. And their DNA becomes a part of the cell's DNA. So this is an E. coli virus. They all have this weird shape called complex. They look like lunar landers. Yeah, that was my generation. We went to the moon. Where have you gone? All right, never mind. All right, so anyway. The bacteriophage, the E. coli, very specific for E. coli, will attach, and then it will inject its DNA, and its DNA will complex with E. coli's and take over the cell and tell the cell you're no longer E. coli. Your job is only to make virus particles. And it will make the virus particles, and then it will go back and it will copy its own DNA to put them in these little virus particles. But when it copies it, sometimes it picks up one or two on either side by accident. So some of these that are released into the environment have one gene or two genes from E. coli. And when they attach to another E. coli, they transfer a gene from one E. coli to another. Now, this is the one that Hershey won his Nobel Prize on. And it was amusing when they first discovered it, that viruses can attack bacteria. I mean, it made us feel pleasantly warm inside that viruses don't just attack us. But it was not until in UCLA in the 90s that it became an idea that maybe we can fix people's genetic damage or inherited disease by carrying the right gene into where it's missing. And one of the first experiments was done at UCLA on cystic fibrosis patients. Cystic fibrosis, one of the kinds of it is you don't make, you remember for your lungs to absorb oxygen, they must be wet with a surfactant. And your body makes it every day. It also has a gene to digest it, otherwise you drown in your own fluid. Cystic fibrosis, some of the cystic fibrosis patients are lacking that gene to digest or to rid the body of that surfactant it makes every day. So they actually have to stick a tube down their throat and pump it out all day long. And if you've ever seen them, they're all pretty deformed and they haven't grown well and they're in a cart and you can hear it going, pumping the liquid out of their lungs. And they don't live very long. So, here's what the scientists at UCLA thought. Why don't we take a virus, a cold virus that infects the respiratory cells, and knock out the part that makes it a pathogen, a cold pathogen, and put in the gene that helps digest this fluid. Then have the patients breathe this recombinant virus, and it will infect the cells of the respiratory system and carry the missing gene. So they did all kinds of amniotic studies and found 5 patients that were going to have babies that were born with cystic fibrosis. They made the recombinant virus, and the moment these babies took their first breath, they breathed in this recombinant virus. 2 of the 5 were cured. They never developed cystic fibrosis. The gene was that they had damaged or was defective was carried and infected their respiratory cells, just like cold viruses do. So this led to the idea, a whole new medical science called gene therapy, that you can fix somebody's genes after they're born. And of course, that means we can get rid of diabetes, we can get rid of inherited cancer, we can get rid of sickle cell anemia, we can get rid of so many inherited genetic diseases. Now, lots of people think that gene therapy is a reality. There have been thousands, tens of thousands of experiments done, and just a handful have been successful. So they don't know all the rules yet. But they have cured a certain kind of blindness. There is a liver defect where certain people don't make a certain enzyme, liver enzyme, that they've cured this way, but not a lot. And let's talk about why. This is just what it is, not important. Okay, so let's go on here. Scientifically, this was moderately important. To discover a virus covered infected bacteria was nice. It didn't become major until the 90s. So it was known in 1952, but it wasn't important until the 90s. Psychologically, now, it is tremendously important because it created a whole new medical science of gene therapy. All right, remember, the original gene therapy experiments, though, are really specific. The E. coli virus will only attach to a certain E. coli. So you have to find a good gene. You've got to find a virus that will carry it to the right place. And the worst problem is you've got to get it in the right spot. All of which are very difficult. So, and of course, since it's only moving one gene, it has little effect on evolution. All right, so let's talk about gene therapy. These are all the things they think they can fix with gene therapy. And of course, very little has been realized because they have a lot of problems. What are the problems? You've got to have a disease that's caused by one defective gene. Many genes working together cause disease. So you have to find a disease that only has one gene. It's a problem. Then you've got to find a good one. You know, a lot of your genes are only partially turned on. Now, you've got to carry the gene to the right cell. And so they tried retroviral vectors, and they tried fat droplets. You know what they do now? They inject it directly into the cell. Naked plasma DNA. So these were all, you know, at the beginning, they used vectors, other viruses to carry them. Now, they just inject thousands of copies into the cell. Okay, so gene therapy, what are the problems with it? One, you got to find a genetic illness that's caused by one defective gene. You got to find a good gene. You got to find a vector that will carry it into the host cell and put it in the right place. So there's 6 problems. And by the way, there have been disasters. For instance, they found a gene on HIV that inhibits replication. So, remember, SIV and HIV are similar. SIV is simian immunodeficiency virus, and it's in AIDS. So they used gene therapy to make a whole bunch of copies of this one gene that stops HIV from replication. And they got it carried into T cells, except it inserted in the wrong place, and the cells reproduced at 100 million times faster than they did before, and it killed them in 3 days. So getting it in the wrong place is a big problem. They were doing gene therapy on 2 little kids that had a minor immune system function wrong. And they used gene therapy, and they both ended up with leukemia and had to go through leukemia treatment because the gene went in the wrong place. So there have been lots of failures with gene therapy, and there's a lot we need to know. Okay, the last of the big naturals is conjugation. Sex. You saw that bacteria in the movie having a three-way, right? Okay. There's three of them with their little pili all intertwined, like the 60s. Well, anyway.
What makes the
What's the name of this video? Dr.
I'm seeing life on Earth.
Nope, that's the series.
Gene transfer.
I wrote it on the board, I said it 5 times. Think about it.
Can you just pull it up on your site on your YouTube?
Yeah, but it's really small and not so good, but I can get this going. I have one very good and very bad trait. One is I never give up, ever.
We're watching this, and then we're also watching the
Yeah.
mouse that landed go.
But not at one right after the other, we're going to go right through it. Now it starts, when I threaten to throw it on the floor. Okay.
Dr. Hicks, is there,
Down deeper, all of you.
Is there what?
never mind.
And by the Office of Biological and Environmental Research of the United States Department of
And conjugation. And we'll also learn how humans have harnessed these mechanisms and applied them for the benefit of all of us. Unlike you and I, or any other creature you can see here at the fair, microbes can pass their genes not only to their offspring, but also laterally to other microbes of the same generation. This is known as horizontal gene transmission, and it's a trait that really sets the microbes apart from multi-celled life.
More than one pilot.
To learn more about horizontal gene transmission, I'm here at Oregon Health Sciences University to meet with Dr. Maggie So, chair of the university's Molecular Microbiology and Immunology Department.
I'm interested in how these microbes can acquire virulence traits. And many of these characteristics can be acquired, can be rapidly spread among a population of bacteria through these mechanisms. The microbes in your body might be totally harmless until they acquire these traits through horizontal transmission. And by acquiring these traits, they can suddenly become more virulent. They can acquire drug resistance, for instance. So if they cause disease later, they would be harder to treat.
So is there a disease that's a good example of this horizontal transfer process?
Well, a good example of this is diphtheria, which is a disease we don't see anymore because of childhood immunization programs. The disease is caused by a bacterium called Corynebacterium. And for the bacterium to cause disease, it has to produce a toxin, which kills the cells in your body. And the gene for the toxin is transmitted to the bacterium via a bacteriophage. And a lot of toxin genes
of energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
It's no secret that genetic information is passed on from generation to generation. I mean, take a look around. The passing on of DNA in plants and animals is achieved by a mixing of genetic information, which occurs as a result of reproduction. And it's easy to see the genetic traits that organisms share. Microbes pass on genetic information too. While their sex lives are quite different from ours, their DNA is nonetheless successfully regenerated. Beings, along with other complex life forms, transmit their genes only to subsequent generations. So-called vertical gene transmission means that genetic information is passed on only through the reproductive process. Microbes do this too, but in addition, they have another extraordinary ability that is a far more powerful transmitter of genetic traits. I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore the three main mechanisms for microbial genetic recombination. That's transformation, transduction,
are actually transferred to bacteria through the process of phage conversion.
Phage conversion can result from transduction. Genetic material passes from one bacterium to another via a bacterial virus called a bacteriophage. The virus injects its DNA into the bacterium. The bacterium then becomes a virus factory, replicating the viral genome, making viral coat proteins, and eventually releasing hundreds of new bacteriophage.
Every once in a while, during replication of the viral genome, a small piece of bacterial DNA gets inserted into the coat protein as well. And the live virus now transfers this genetic information from the recipient bacteria to a new bacteria.
In conjugation, another form of genetic transfer, a donor cell comes in contact with a recipient cell by way of a conjugation tube known as the sex pilus. When contact is made, a single strand of DNA containing the genetic information goes through the pilus from the donor cell to the recipient cell. This single strand of DNA is usually in the form of a plasmid, which is an extra chromosomal, independently replicating piece of DNA. A second strand of the genetic information quickly gets...
Technology now. So, let's see if this works. You know how many times I have trouble with this, so I don't know. Called Gene Transfer. It's in the Unseen Life on Earth series, so you can't see it online if you wish.
It's called gene transfer.
Yeah.
Chapter 7.
I want to throw this thing out the window. Anybody have a window? Okay, I am prepared for this. I have 4 copies. This machine loves giving hell.
I'm not fun about
Now this one needed 8 last year, so at the beginning it was a little. goes in your lab record book, front to back, under Unseen Life on Earth, and transfer, and nothing. Would you hit the VCR button over there again? Nothing, right? There it is. Now it's going fast. Hey.